My presentation is on an article titled Lack of MID1, the mouse ortholog of the Opitz syndrome gene causes abnormal development of the brain. Opitz G BBB syndrome is a genetic condition causing abnormalities along the midline of the body that occurs prenatally. There are two forms of this syndrome. The first form is an X-linked recessive form of inheritance that affects 1 in every 10 to 50,000 men around the world. The reason why men are more so affected is because, as you can see, men only have one X chromosome, and therefore even a recessive mutation will show a phenotype. The second form is an autosomal dominant form. However, this form isn't well understood because it's actually part of a larger syndrome called 22q11.2 deletion syndrome, and therefore it's very much underdiagnosed. Although these two different forms have different inheritance patterns, they actually present the exact same symptoms, which I will talk about later on. The way that this disorder presents itself phenotypically is by defects in the formation of structures along the midline of the body. Now, these defects can vary between different patients, but it often results in things such as ocular hypotelorism, which is wide-spaced eyes. There are also defects in the larynx, trachea, and esophagus experienced by most, but some people will have something called a laryngeal cleft, which is a hole that forms between the larynx and the esophagus, which makes it very easy for food to pass directly into the windpipe and therefore can be life-threatening. There are also genital abnormalities, such as infertility, or some men will experience a misplaced urethra or a separated scrotum. And then in certain patients, they experience an intellectual or developmental delay. This is a result of the structures of the brain not being able to form properly, which I'll talk about later on. And then another 50% of patients will see a cleft lip or a cleft palate that forms. The image shows two brothers, both of which have Opitz G BBB syndrome, and you can see that they also have widow's peaks and a lot of different structural and cranial abnormalities I didn't mention. This is because, as I mentioned before, the phenotype will vary. So why is this disorder caused? Well, it's due to defects in the gene called MID1. Now, in Opitz G BBB specifically, there can be up to 90 different mutations in this one gene, which is what causes the huge variability in symptoms that we see. I've put an image of the X chromosome where MID1 can be found, and it's indicated by a yellow arrow. Now, MID1 forms a protein called midline 1, and this protein is what helps cells migrate along the midline during development. And as we know from learning in class, the migration of cells is crucial to form structures in the developing embryo. Midline 1 specifically binds microtubules and recycles proteins, and so when there's a mutation that disrupts the function of midline 1, that will result in cell buildup, depleting microtubule function, and this causes improper cell migration, which will have an effect on the structure. I put an image of a fish from the lectures we've learned, and you can just see that in these fish, migration later on in development is crucial to form the structures of the fish. And this just goes to show how important migration of cells is throughout all organisms. In terms of diagnosing Opitz G, it's normally diagnosed by just the physical symptoms. However, a lot of the features can't be distinguished from other syndromes, such as 22q11.2 deletion syndrome I mentioned before, which is why this disorder is often really underdiagnosed, especially when it's in that autosomal dominant form of inheritance. Nonetheless, genetic testing is done for people who do show physical symptoms, and also for people who might have a family inheritance of it. They test specifically for the mutation in MID1. Right now, there are no genetic treatments or therapies for this mutation. There are only treatments for the physical symptoms that arise from the mutation. So, for example, surgery can be done for a variety of different features. In this image, I've included a child who has undergone a cleft lip surgery, which is very easy to do. In advance, things can be done such as speech therapy or intellectual therapy for patients that might need it as well. So the overall research question is, how does the MID1 gene mutation found in Opitz G BBB disorder affect mice behavior and brain development in vivo, and how can findings be used for future studies into the gene's involvement in brain function? The way scientists got to this is because at the time, MID1 was known to be involved in this disorder. However, its pathogenesis, especially in vivo, was not well understood. In advance, they were very much aware of the clinical features, but they didn't know what was really happening in terms of brain pathogenesis and Opitz G BBB. So the goal here was to develop a MID1 knockout mice to study the effects of Opitz G in vivo, specifically in terms of brain development and behavior of the mice, so they could hopefully one day translate that into other organisms. How they went about answering this research question was through their experimental design, where they essentially used CRISPR to generate MID1 knockout mice that had the Opitz G disorder. They took wild type and these knockout mice and they subjected them both to behavioral analysis. They did so with 90 mice, half of each. They then took the tissues, the brain tissues of these mice, and prepped them so they could go through RNA in situ hybridization and immunohistochemistry to actually be able to visualize the differences between all their brains and the differences between the RNA mutations within their brains. All the mice were bred for 10 generations. However, I should mention there was no mention of how many mice that were used in specifically tissue sampling, as well as no mention of any other controls apart from the wild type and the knockout mice. So the first part of the methods is the biological methods, where they use cryosectioning to prep the brain tissue from the adult male mice and subjected it to RNA in situ hybridization to detect the mutations of this tissue. Lastly, they use immunohistochemistry for tissue imaging, which is, as we all know, is crucial in developmental biology. That's why I've put the process of it to the right. Essentially what it is detecting a certain protein by binding antibodies that are able to recognize this protein, and then binding a substrate which will release a colored product so we can view the image. Then there's the behavioral methods. So what they did here is they studied 45 wild type and 45 knockout mice, all of which were male, and all of which had consistent living conditions. They tested firstly motor coordination and then learning and memory. They tested motor coordination by having the mice walk on steel wire for 120 seconds and just seeing how many times they would fall off. Then they tested learning and memory over the span of a few days. They tested motor learning using something called the rotarod test over the course of 5 days. I put an image of what the rotarod test essentially consists of. It's a rotating rod which the mice are placed on, and it tests their motor ability to stay on this rod. Then, their memory was tested using a food maze over the course of 15 days. Food mazes are used a lot in mice studies. Essentially, they put food in the middle of a maze and they put the mice in this maze. Over the course of 15 days, the mice will repeatedly be put in this maze, and they test to see how well the mice remember the exact path to take to get to the food. Lastly, a statistical analysis was done using ANOVA. The results showed that there were a lot of brain formation defects that all arose prenatally. This caused improper brain definition, and this was all due to a lack of cell migration. This caused problems like lobe defects, fissures, mispositioning of certain brain boundaries, but scientists here only focused on certain areas of the brain. The first area, as you can see in image 1, is the cerebellum. Here, the lobe formation was improper. As you can see by the arrow indicated, there wasn't the right number of lobes that formed when the mice were knockout. There was also an improper number of granule cells and dendrites within this area. This will obviously have huge implications into the function of the cerebellum in these mice. The second place they looked at was the anterobasal lobe. Here, the granule cells actually migrated too far inwards. This caused fissures within the lobes. These findings obviously mean that there are a lot of different brain formation defects in a lot of different areas of the mice brains. Another area they looked at was the midbrain and the hindbrain defects. This is very interesting because the midbrain and hindbrain is actually established by fibroblast growth factors, FGFs, which we studied in class. They saw using the RNA in situ hybridization that MID1 decreased FGF17 expression in these mice. What this meant is that the isthmus, which is the area in the brain that separates the midbrain from the hindbrain, was actually shorter in length and completely mispositioned, as you can see in image A, B, and the arrows indicated. This caused a larger boundary between the dorsal midbrain and the cerebellum hindbrain. The thing I thought was really interesting is that because MID1 regulates FGF expression in mice, they actually found that it regulates FGF8 expression in chicks. The image below shows how FGF forms in the Henson's node and is important for the formation of the primitive streak, the notochord, and the brainstem of a chicken. I thought this was really important in mentioning how these results can be used in other organisms as well as just mice. And then lastly, we have the behavioral results. Scientists here saw that MID1 knockout mice have impairments in motor coordination and in learning. When they did the rotarod test, they had a higher form of latency, and in the maze, they just weren't able to get the correct response as much. They think this is likely due to cerebellum defects because the cerebellum is responsible for spatial awareness, for motor coordination, and so on. So obviously, the formation of the cerebellum wasn't correct, and therefore, the behavioral response will not be correct either. So to conclude, MID1 mutation Opitz G BBB does cause brain and behavioral abnormalities in mice and can be used as an animal model for other organisms. However, it should be noted that mice and humans Opitz G or MID1 knockout is very, very different. As I mentioned before, a lot of humans are infertile, their life can be threatened by this disorder. However, in mice, they all had a normal lifespan, they were all fertile, and they didn't show symptoms like things like cleft lip and physical symptoms like that. So for that reason, it's very important that this research can only really be translated in terms of brain and behavioral effects. The next steps would probably be to look at the mid-1 pathways in humans and chickens and other organisms and see the implications for comparing these brains to the mice brains studied in this analysis. Personally, I thought it was overall a really, really well-written, detailed, thorough researched article. I just did think that they should have mentioned more so into the mice and the controls they used, as well as I think they could have improved on discussing some alternative and unexpected findings because they didn't mention any, and I thought as a reader it could make my beliefs in this research article a bit biased. Thank you so much for listening to my presentation, and I've included my references here.